Hello everyone. Today we'll be discussing about the gram staining technique. So the gram staining technique, it was firstly developed by Christian Gram, and this one is basically to differentiate different types of bacteria depending on the nature of the color. So this step, it takes place in different ways. The first one is to prepare the bacterial smear. So how is the bacterial smear prepared? The bacterial smear is prepared by basically taking a slide. We are taking a glass slide, and there will be adding a drop of distilled water, just a tiny little drop of distilled water, and after which there will be a culture plate from of bacteria, and from which we will just inoculate a bit of that extract, and then we will put it on the slip containing the distilled water. And after mixing very thoroughly with the help of the inoculation loop on the drop of water, we will leave it for us for a couple of minutes so that it can air dry. And after it has air dried, we will heat fix it. So basically that's the way we will be preparing the bacterial smear. Now for the gram staining part, we will be adding crystal violet to that slip. Alright, so after adding the crystal violet to it, we'll be washing it thoroughly. Not exactly thoroughly, but through running water. We'll be putting that slide carefully under the running water in such a way that the entire smear has not been affected. Only the dye has been washed off. After which we'll be adding the grams iodine, and after grams iodine we will be adding a decolorizer. So in this case we will be using ethanol, that is the alcohol. So it will basically degrade the color that was added before. And after that we will be counter staining it. Counter staining means that we will be adding a second dye. The first dye was basically the crystal violet, and now the second dye that we'll be using would be a red colored dye, which is the saffronine. After that we'll be washing it off, and after washing it off we will observe it. So if it appears under the microscope purple color, we can say that it is a positive bacteria, a gram positive bacteria. But if it appears negative, if it appears red in color, it is a negative gram bacteria. So that was all for today. Thank you very much.